Class, we're going to continue our chapter on genetics. We'll be continuing with our discussion of the translation step in protein synthesis and move into a discussion of the genetic code, the universal genetic code. Where we left off was a discussion of how messenger RNA is synthesized by making a copy of the strands of DNA. The messenger RNA travels to the ribosomes where it is attached, and then we have a process of stringing the amino acids together to form a protein. That process involves another RNA molecule called transfer RNA, tRNA. And shown in this slide is a structure of one of the tRNAs. Now, there are many, many different tRNAs for reasons we'll see in a second. But they all have a very similar structure as shown here. It's kind of like an L-shaped molecule, but there are two really important ends to the molecule. On this top part of the molecule is where one of the 20 amino acids is attached covalently. That is, it's bonded to a position here. And the amino acid will then react with the growing string of amino acids, the growing polypeptide, to extend that polypeptide by one more amino acid. So essentially this amino acid will become attached to the growing polypeptide chain in the process. The other important end of the molecule is what is called the anticodon loop. This anticodon loop has three nucleotides that turn out to be complementary to the codon, the sequence of three nucleotides on the messenger RNA. Remember, we said that the genetic information is in terms of a three nucleotide sequence called codons, which we'll talk a little bit more about in a second. And that the three codons code for one of the 20 amino acids. Again, we're talking about a code from one language to another, essentially. And the part of the code reading process is that we have an anticodon set of three nucleotides at the bottom of transfer RNA. Those anticodon nucleotides are complementary to the codons in the messenger RNA. That is the way they line up so that we get the proper amino acid being attached in the growing chain. This slide shows what is called the genetic code. I say it's a universal genetic code because it is used by essentially all life forms on planet Earth, from the most ancient bacteria to humans and to plants and bacteria. It's used by all life forms with very, very rare exceptions, and with those exceptions, it's very, very small exceptions to this universal genetic code. Now, what I show in the table are 64 different codons. Again, a codon is a three nucleotide sequence. So the very first one in the table is UUU. That is when three nucleotides together, three uridines together, uridine, 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 right in a sequence. Those three nucleotides code for an amino acid called phenylalanine. Now, PHE is a biochemist abbreviation for the amino acid phenylalanine. There are 20 of these different amino acids. So if we look at the amino acids, the three-letter abbreviations for these, if we were to count these up, there would be 20 different amino acids. There are 64 different codons, 64 different three nucleotide sequences. And so you'll see that there are two codons for phenylalanine. Leucine hit the jackpot. There are 6 codons that will code for this amino acid called leucine. There are 4 for serine, 4 for proline, 4 for threonine, 4 for alanine, etc. Many of them have 2, like tyrosine, TYR, has 2 nucleotides. So either there usually are 2 or 4. In some cases, there are as many as 6. And this one, tryptophan, has only a single codon that will code for the amino acid tryptophan. The point is that there are these codons that are codes for delivering a particular amino acid next in the sequence of the growing polypeptide chain. Now, it's kind of interesting in that we have more codons than you would think are necessary. We have 64 codons. We only needed 20. But what is interesting is that the third position seems to have a lot of play in it. That is, if you'll notice UUU or UUC, that is a variation in the third codon, doesn't make any difference. It still codes for phenylalanine. If we look at serine, the first two nucleotides have to be U and C. The third one could be any of the four possibilities. So there's a lot of play in the third position, and that has led some people to think that maybe originally this genetic code involved only two nucleotides together, and maybe there was a spacer, a comma, for the third position. But over time, that third codon became important to specify the growing number of amino acids used in making proteins. But the point is there are 64 codons for 20 amino acids. There's some redundancy in that we have more than one codon for most of the amino acids. All life forms use these 20 amino acids, etc. So we call it the universal genetic code. There are a lot of interesting features about this, but that's for biochemistry nerds like me. Okay, so how does this work? We've talked about all the important components. This slide shows a ribosome. This tan-looking blob thing is supposed to be a ribosome. And a ribosome is made of proteins and RNA, ribosomal RNA, and it is thought to have a couple of important features. One, it has places to bind or to interact with two different tRNAs. These things that you can kind of tell they look like that L-shaped tRNA molecule that we showed in its full glory a couple of slides ago. It has positions to bind two different tRNAs, and also it has a place where it will bind the messenger RNA. This, of course, is our messenger RNA, our sequence of nucleotides, our sequence of 3 nucleotide codons. Okay, so what happens is that the tRNA, having an amino acid already attached to it, will come in and will find a location with the anticodon loop interacting in a complementary manner with the codons in the messenger RNA. When it does so, in the second position, there will also be a transfer RNA with a polypeptide chain that has already been there and already is growing. And this polypeptide chain is transferred to the incoming transfer RNA to grow the polypeptide chain. It then kicks out the naked or the spent transfer RNA, and the transfer RNA with the growing polypeptide chain moves over into the second position, leaving space for yet another transfer RNA to come in, and the one that comes in has to be one that is complementary to the nucleotide sequence in the messenger RNA at the bottom, and everything sort of moves in one direction. That kind of zips through as you go. And in this way, we essentially grow a polypeptide chain till we get to an end stopping point. Now, typically, a growing polypeptide chain, what will become a protein, might be 100 to 200 amino acids in length. That's kind of like an average length. Once a polypeptide chain has been synthesized, we think that initially it is unstructured, just a sort of a random polymer. But proteins have this interesting ability. Most of them have this ability by themselves to fold up into a three-dimensional structure. A three-dimensional structure which has some biological function. It might be an enzyme, it might be an antibody molecule, it might be something like hemoglobin, which binds oxygen, it might be a structural protein, it might be a hormone like insulin. But the point is that the proteins are functional molecules. They're almost like little machines that will do something. And many polypeptide chains have this ability to fold up on their own to form this functional three-dimensional structure. And in some cases where they will need assistance from something that is called a chaperone protein to help them fold. In some cases, the protein has multiple subunits, so it has to be joined together with other subunits. Like in hemoglobin, there are actually 4 subunits. So that is how proteins are made. Now, in this last slide in this lecture, I'm just going to throw out some contemporary topics in molecular genetics. I won't have time to talk about any of these in depth, but first of all, let me say something about mutations. DNA and the replication of DNA, the transcription of DNA, and the translation step, all of these things are performed by molecules, and molecules are not 100% perfect. I mean, they're chemical machines, but they're also susceptible to chemistry. That is, other things can react with them. They can react with chemicals in the environment or ultraviolet light, things that can damage or mess up or just react with the nucleic acids, with the proteins, with the enzymes that are catalyzing all of these reactions, leading to mutations or errors. And so that turns out to be something that is important. That is, all of this doesn't operate perfectly. It operates with very high efficiency and with few mistakes, but with some mistakes, and some of these mistakes can lead to mutations. And in a later chapter, we'll talk about how mutations are actually important in enabling evolution. Sometimes the mutations can be harmful, sometimes they can be beneficial. Now, even though mutations occur, there are enzymes in our bodies that attempt to repair these mutations. One of the most well-known examples is damage to DNA caused by ultraviolet light, forming something called thymine dimers. It turns out that our cells have an enzyme that goes around and looks for these types of photochemically induced mutations in DNA and snips them out and lays down fresh nucleotides that essentially to repair the errors. Now, the problem is, what if that repair mechanism gets swamped and can't keep up? And that's what happens in some cases that lead to cancer, especially skin cancer caused by ultraviolet light. Another topic that, if we had more time, we would talk about is the control of the expression of genetic information. All of our cells have the same genetic information. That is, our liver cells, our muscle cells, our nerve cells, our bone cells, our epithelial cells, all have the same genetic information, but yet all of these cells are different because different proteins have been expressed. In other words, different genes have been expressed to make proteins that then determine the nature of that particular cell. In muscle cells, we make these proteins called actin and myosin, which are important in muscle contraction. In nerve cells, we will make enzymes that are involved in the synthesis of neurotransmitters. So we have different cells because different genes have been expressed. Well, how do we control all of this, turning on and off the expression of genes in different cells? That's what this topic is all about, and it's a lot of people doing a lot of research studying this, and it's a fascinating field. Let me skip to this Human Genome Project. This was one of these moonshot ideas that started around 1990, where the idea was, wow, if we could just determine the sequence of all of the chromosomes in a human and in other organisms, maybe we could then understand the genetic basis of diseases and of variations between humans, and we would understand the genes for various proteins. And so that project was initiated around 1990, and it was thought at the time that this might take forever, but in about 12 years, they successfully sequenced the entire human genome. It's one of the great success stories in science. What they learned, surprisingly, is that there's a lot of what they call junk DNA, that is, parts of the chromosomes, that is, sequences of nucleotide in the DNA, for which there is no obvious function. That is, there were not genes. They didn't appear to code for proteins. Now, scientists are continuing to study this junk DNA, and they're finding some functions for some, but as much as 95% seems to not be DNA that is actually part of a gene per se. And it's lumped into this category called junk DNA, which basically means we got to study this more. We don't know if it's really just junk that's been carried around, just like we have in some of our closets, or if there is some function and we just don't know what that function is yet. Now, another topic is the genetic basis for disease. I said that one of the reasons for wanting to determine the sequence of nucleotides in the entire human genome, all of the human chromosomes, was to try to develop a database so that might help us understand the genetic basis for diseases. And in fact, there's a lot of research being done in that area now. Some genetic diseases, for example, are cystic fibrosis, sickle cell anemia, colored blindness, Tay-Sachs disease, Down syndrome, PKU disease. These are some rather well-known genetic diseases, and in many cases, scientists are determining exactly what the genetic basis is for these diseases. Sickle cell anemia was one of the ones that was understood very early on. It's due to a mutation in a single place in hemoglobin. Now, if you look at the backdrop, you'll see you might recognize that as being something that looks like either a barcode, or you might be aware that these are images like those prepared when sequencing DNA or determining DNA fingerprinting profiles. By DNA fingerprinting, we mean determining the location of various what are called markers on chromosomes, and that helps us identify individual human beings. That is, DNA from different individuals has uniqueness, and we will have different genetic markers at different locations, and there are ways to visualize the location of these markers using this technique of DNA fingerprinting. Another final topic just to mention is gene replacement therapy. This sort of goes along with the genetic basis for disease. An active area of research is to try to develop ways to actually replace non-functional genes. That is, if you have a genetic deficiency disease because the gene for a particular important protein just isn't working, the idea is to try to replace it by gene replacement therapy. So that's something that is a hot area of biomedical research, and you might hear of a technique that has been developed only in the past couple of years called CRISPR, CRISPR. If you hear somebody talk about CRISPR, it's like a hot new technique used to sort of snip in and out and do editing of DNA as a way to assist in gene replacement therapy. So that's something new. Maybe as time goes on, we'll have the ability to treat a number of these genetic diseases by gene replacement therapy. Okay, we'll stop again and have a little quiz, and then the next topic I'll talk about, because it's in the news, of course, viruses, and we'll talk about what a virus is and give several examples of viruses. Okay?